Good evening to everyone. Good evening to everyone. I welcome you all to today's ENVIS webinar topic, Diatoms of Ocean by Sharanya G. Sharanya is PhD student at Center for Sustainable Technology, ASTRA, and also associated with Energy and Wetland Research Group at Center for Ecological Sciences at Indian Institute of Science. Her research area is focused on developing novel algal bioprocesses from indigenous strains for the economical production of third-generation biofuel. And her other research interests includes extraction of lipase as a sustainable biocatalyst for enhancing biodiesel yield through enzymatic transesterification. And before joining Indian Institute for her PhD program, she has completed her MTech in Energy Engineering specialization from National Institute of Technology, Tiruchirappalli. So over to you, Sharanya.
Thank you, Dipti, for that wonderful introduction about me. So I welcome you all on behalf of Lake Organizing Committee as well as the ENVIS webinar series for this talk. I would like to extend my sincere gratitude and thanks to Dr. T.V. Ramchandra sir for giving me this opportunity to address you all in this webinar and showcase my talent in this platform. So today the session is on diatoms. Especially I will cover the economic as well as the ecological aspects of diatoms. I would like to initiate or start my talk by telling this famous quote by Lauren Aisley, who said, "If there is a magic on this planet, it is contained in water." So, having been worked in this field of coastal ecosystem and diatoms for over 6 years, I strongly concur with this statement. And in this talk, through this talk, I would like to take you through how I reinstate this statement, and also how diatoms are indeed going to be they are like marvelous creatures of the ocean, and how it is going to be the organism for both ecology as well as economical and industrial relevance. So to start with, I'll give a brief introduction about diatoms. So on the left hand side, you see an image. That is how a diatom look. This is a microscopic view of a planktonic diatom. So you can see how diverse the morphological variants are. So it came into existence from some 1,000 years or 100 million years ago. So having said that, you can see the quantum of biomass that has been formed in the ocean. So it became a major organic source for the major marine creatures. So there are hypotheses that even says that diatoms are the one which are responsible for the whales to appear, because there are a quantum of food that is available all throughout the Mesozoic era. So the first life started in the ocean is what one of the debates says. So it has given rise to that gigantic creatures like whales also. So that is the importance this diatom get in an ecological perspective. So and also it is known that 1/5 of the photosynthesis that occurs in the whole planet as such is from these diatoms only. So having said that, it plays a major role in carbon sequestration also. So photosynthesis involves carbon dioxide as well. We all know that. So a large amount of carbon dioxide is getting sequestered because of this diatoms, and also they are known as the savior of the marine life as such. So having said that, this is the morphological feature. So you can see a centric diatom here. So it is typical. It has a typical pillbox structure. So it has an epivalve and hypovalve, which is joined by these griddle bands. This is the morphological at a macroscopic level. This is the morphology that the diatom possesses. So based on this, many nanostructure prototypes also is created. What you see in the right hand side is the prototype of a nano scale structure that got inspired by these diatoms. So moving on, I will take you through this contents. So I will start with introduction, which will cover the morphology as well as the ecological perspective of how it came into existence, and what are all the importance of diatoms in the in an ecological sense, as well as I'll take you through the diatom sampling. The sampling aspects will involve, if it is a planktonic diatom, how do you have to sample? If it is a benthic diatom, how do you have to sample? I will explain in detail what are these planktonic and benthic forms in detail in my further slides. And then I will take you through diatom blooms, how it is causing a menace during this days because of the anthropogenic pollution that we create. And also diatoms as bioindicators. So nowadays these are being used as a tool for monitoring the ecosystem as well, aquatic ecosystem as well. And then I'll brief you through this diatoms on the west and east coast, what I have collated the information on. And then I will start on the industrial aspects of or the economic benefits of these diatoms as a and I will not go in detail of it. I will cover this, but all these after this diatoms along the west and east coast are the industrial aspects of diatoms. I will take you through in detail on all of these aspects. So diatoms, these forms are basis of the food web. So in marine ecosystem, if you consider, so these are the primary producers. It is, if not, they are the major primary producers. It shares a good amount of biomass or the population with seaweed, sea grasses, and kelps and other microalgae. So these become the food source for the higher organisms. So that is how the food web continues. And also from the carbon sequestration side, micro and macro algae are the major carbon sequesters, which is contributing to nearly 40% of the carbon fixation that is happening. So that is, you can see the carbon fixation by the terrestrial plants is just 3 to 6%. From this you can understand the quantum of work that these microscopic marvels are doing in terms of making us alive in this planet, because if not for these diatoms we would have been dead by now, because the carbon dioxide pollution or the carbon dioxide concentration in the atmosphere is getting increased day by day. So it is because of these organisms we are able to survive, because it makes the planet livable in some sense. So where these diatoms actually fall? So in the kingdom of five classification kingdom levels, so we have bacteria from animals. It starts with bacteria and ends with animals. So the diatoms fall in this algal category. So in this algal category, algae are basically divided into macro and micro algae. So and they are categorized based on their pigments. So you have red, green, and brown algae based on the type of pigments that it secretes. So is diatoms being a part of this red, green, and brown algae? If you ask, so it is not so. So diatoms are categorized into a different group in itself. Why? Because they, the cell structures and the properties that it holds, and also the pigments that it secretes is well distinguished from all of these other microalgae. So that is why diatoms are placed in a different position than other micro like green and say for example green algae and cyanobacterial species. So having said that, I'll move to the next peculiar characteristics of diatom that distinguishes it from the green microalgae. So it is a silica cell wall. You can see here on the right side, you can see here these are transparent glass-like structures. So silica, you know, it is a basic component of a glass. So glass is also made up of silica, and it is a different crystallographic structure of a silica itself. So you can see here these are transparent glass walls, inside which you can see some fluidy brown golden brown fluidy substances. So this is very transparent. You can see what is inside the cell actually. So that is why the diatoms are popularly known as algae in glass houses. And also it is characterized into a separate category because of this golden brown pigments. It is because of the secretion of xanthophyll. Xanthophyll is again a pigment which is categorized into carotenoids. Carotenoids is again the photoprotective pigments. They are called as photoprotective pigments. So the xanthophyll is a subgroup of this carotenoids. So in carotenoids you have many different carotenoids: beta carotene, lutein, and xanthophylls. So diadoxanthin and diadenosanthin, these are the two peculiar xanthophylls that are present in diatoms. That makes these organisms very peculiar. And also, if you see green microalgae and diatoms, green microalgae has a major composition of chlorophyll A and B, whereas this diatoms forms a major composition of chlorophyll C. The chlorophyll C is at a higher level. So why is it important? Because the absorption of this electromagnetic spectrum from that is from the sun. So because these are photosynthesizing organisms, what happens when the sun rays fall into it? So the level of wavelengths that it can absorb varies based on this chlorophyll A and C, and also the color that it reflects is a proxy of what chlorophyll it actually has. So green algae, why it is green? Because it absorbs all the other wavelengths except for green. So similarly, why diatoms appear in golden brown color? It is because of this why golden brown? Because it is because of the wavelengths that it absorbs and it reflects. So it absorbs all the wavelengths except for this orangish yellow wavelengths of the electromagnetic spectrum that is coming from the sun. So that helps them to photosynthesize efficiently. And also, yet another use of these carotenoid pigments is that because diatoms are predominant in the ocean surfaces receives a huge amount of sun rays. So you can see that it is not only the photosynthetically active region, that is from 400 to 700. The electromagnetic spectrum consists of even the harmful UV rays also. So UV rays falls into this ocean surfaces, and it directly gets absorbed during the photosynthesis. It cannot it doesn't have a screening mechanism before that, so it absorbs all the spectrum. So what happens if the long wave radiation or the harmful radiation passes into this structures, it will hamper or impair the photosystems, and it will not be able to photosynthesize effectively. So it damages the photosystems and in the chlorophyll pigments, which is again present in the thylakoid membranes. So that is why the carotenoids are present. So carotenoids, what happens, like you would have seen in green plants that is happening. So excess amount of radiation, solar radiation is there. You can see some transpiration, that water droplets present in the leaves, that is called as transpiration. So the same way, what it does, the extra radiation which is not useful for photosynthesis are absorbed through this carotenoid pigments, and it transpires it in a way. It doesn't allow it to the reaction center. It diverts or reroutes that into the non-radiative dissipation, that is called as non-photochemical quenching. So the quenching this process doesn't yield to the any like production of carbohydrates or food sources. Instead, the excess amount of solar radiation which is going to damage the photosystem is getting like avoided through this carotenoid pigments. This is one peculiar characteristic feature of this diatom. This is why the carotenoid pigments is been it is the much sought pigment, I would say, because you have in cosmetic industries, you have many UV protective layers coming up, the cosmetics coming up, because we are exposed to a lot of UV rays nowadays because of the ozone depletion. So this cosmetic industry is looking for such carotenoid pigments, cheap carotenoid pigments. So diatoms becomes the first cheap source of getting it, because producing it is not going to be very tough. So that is why the major cosmetic and pharmaceutical companies are back of this diatoms, just for this one particular pigment that it secretes. And moving on to habitats, where and all these diatoms are predominantly present. As I was telling, the diatoms are present majorly in the oceans. So any water surface is the common habitat for a diatom. But nevertheless, any moist surfaces or soils are going to be the habitat for this. So it is known to have a quick establishment time. So when you are if you want a diatom to get established in any water surfaces, if a water body is nearby, if you are putting any artificial substrata like plastic or wood log also, like a new wood log which is not exposed to moisture before, if you are putting that, within 3 weeks of span, you can see some slimy layer to it. If you scrape that and observe it under microscope, you can see this diatoms proliferatingly growing on this surfaces, and that is how quick it is and swift it is in creating its habitat. So any moist surfaces are going to be the ideal habitat for these diatoms. And also they are some are free floating and some are planktonic, attached benthic forms. So free floating are otherwise known as planktonic phytoplanktons or planktonic forms. So I'll explain these two like types of diatoms in detail in my further slides. And also, apart from that, these are normally available diatoms. Other than that, there are halophytic diatoms also. So those diatoms thrive in a very like extreme conditions. They are called as extremophiles also. So these diatoms, including polar icy regions or the thermal vents also, and hypersaline lakes also, you can find these diatoms. So they are evolutionarily adapted to sustain and establishes its colonies in the extreme conditions as well. Typical examples of these halophytic diatoms include Nischia supralittora and Craticula halophylla and Nischia thermaloids. So these diatoms are like known for their survival or occurrence in this halophytic or extremophilic diatoms, which can be seen in extreme weather conditions. So coming on to morphology, as I was telling, there are two different categories. One is centric ones. They are bilateral, like you can see in the left side, it is a coin-like structure. So you can see some like several pores to it. So they are radially symmetrical. So they are circular in form and they are radially symmetrical. The other one is a bilaterally symmetrical form, and it is called as pinnate diatoms. So these pinnate diatoms are only if you cut into two halves, only in one way you can cut this. So that is why it is called bilaterally symmetrical. So coming on to the in detail about the parts of these diatoms, I'm not going through all of the like cell components. So each has its own purpose. But the one typical characteristics that you have to understand is that it has punctures or pores. If these punctures or pores are linearly or the fashion in which they are arranged, that becomes striae. So linear arrangement of this puncta becomes striae. They are called striae. So why is this important? Because these decides the while you are going for identification of any diatom, these are going to be the major key factors where you identify a diatom based on how the striaes are arranged. So whether it is crisscross or whether it is linearly arranged like that, you will go on categorizing these diatoms or go for classification on which family it belongs and what is the genus name and what is the species name. If you want to go to that taxonomical classification, you need to understand how the striaes appear. So moving on, I would like to talk about the diatom multiplication as well. So this diatom multiplication is a unique way of multiplication or the cell division that you will never find in any other green or any other microalgae for that matter. So as I was telling, it is made up of silica structures. So you have epivalve and a hypovalve. So as and when at a very favorable condition, when all the nutrients are intact, the solar radiation is perfect, at that time it opts for asexual or mitotic cell division, wherein you have epivalve and hypovalve. One parent cell divides into two daughter cells. You can see here, and in the daughter cells, one gets the epivalve and one gets the hypovalve. Alternatively, this keeps happening to the for the generations to come. So as this is happening, for every generation passes, there is a deterioration or minimization in the size. So it keeps reducing in size as and when the generations carry forward. So once it reaches the critical minimum stage where there is no more possibility of cell division, at that time what happens, it triggers the sexual reproduction when the bigger size cells forms the oocytes or eggs, and the smaller size, the smallest of the smallest size, that becomes a male gamete. So the fusion happens, and then after the sexual reproduction gets over, another very important process called oxosporulation happens. It is like you are blowing a balloon, so it blows, right? So the same way, the smaller organism gets blown off to its size equivalent to that of the ancestral parent. So it becomes this process is known as the size restitution. So size reduction happens during the asexual or mitotic division, wherein in the sexual reproduction, oxosporulation is a process that makes the diatoms to come back to its original position or the original shape. So and then once the original shape is regained, it will go back to the asexual reproduction. So this is one peculiar size reduction and size restitution cycle that you will never be able to come across in any other family of algae other than this diatom. So moving forward, I'll give you a glimpse of how the planktonic diatoms in the oceans are. So in the ocean, as I was telling, planktonic diatoms doesn't have a free will to move on its own, because it doesn't have any mechanized structures to it, so that it can flow as on its own free will. It doesn't happen. So what happens as and when the currents, wherever the currents take them, so it has to go. So the colony formation becomes very difficult. But over the course of time, the picture you see here is a coin shaped structures. No, these are all standalone structures of one particular diatom. You can see here. So but colony formation is highly unlikely during such when the water currents are very rigorous. You will not be able to form colonies. So these organisms have adapted itself to form colonies in this way. So you can see here, it is a centipede kind of a structure. This diatom is called a Chaetoceros species. So this is a proper marine diatom, planktonic diatom. So as and when it drifts, it needs to form a colony, so that it can have that colony effect. As you know, all the organisms thrive in colonies. It doesn't thrive standalone. So for forming colonies, these have got a evolutionary adaptation, wherein this flagella kind of structures, these are pseudoflagellates that they have formed, and then these are single structures that are formed together. It might seem to be a centipede, which is an organism itself, but it is a colony of our Chaetoceros species that drifts into the water column. So this is how they stay intact. And also it is considered as a defensive mechanism against fish feeding. So what happens if it is a single cell? So it becomes a favorable food for small fishes. So but for as a defensive mechanism, so it has formed these colonies. If the colony is so big, so the small fishes cannot feed on it. So it can escape being prey to the predator. So that this is this has two purpose. One is for colony formation, the other is to prevent the predator from consuming it. So this is a very beautiful adaptation of this planktonic diatoms, which you can see, which you cannot see in any other planktonic species. And also in the same photograph here, you can see chitin members, chitin fibers. So these are not flagella. You should not be mistaken for this flagella or pseudoflagella, which is shown here. These are chitin species. This helps these planktonic diatoms to float seamlessly. So if the water current is that way, it will align all these chitin fibers to an extent that in an opposite direction, so that it can progress or drift through that wave current seamlessly. For that, it has adapted this chitin fibers. So these are peculiar features of this planktonic diatoms. So and also, as I was telling, the diatoms don't have a free will to float on its own. It has to have it drifts wherever the water column goes. And also I was telling that these diatoms have silicate cell walls. Okay, silica is a denser particle. You can see if you take a tumbler of water and you put sand to it. Sand is nothing but silica. So silica, what happens? It goes down, right? Because the specific gravity of, or the density of silica is much higher than water. That is why it sinks. It immediately goes and sits down in the bottom of the tumbler, right? So that's being said, this also is made up of silica. So you can see here, its natural tendency of any diatom is to go down. right? But for photosynthesizing, at the bottom of the river or the ocean, you will not find the sun rays to come enter. So the solar irradiation cannot reach the bottom of the river or the ocean. So that is why it has to float. It has to keep itself in the euphotic zone, so on the pelagic surfaces of the ocean, so that it need not go down. So, and it need not get deprived of the solar radiation that is going to help them in photosynthesizing. So that is why these diatoms have acquired the adaptation of secreting oil. So you know that if you pour oil in a water, it is going to obviously float, because oil is having a specific gravity which is lighter than which is lesser than the water. So that is why the oil floats in any if you are mixing oil with water, oil is gonna float. So it is the same mechanism that these diatoms have evolutionary adapted. And the planktonic diatoms are known to secrete a lot of oil. So oils are nothing but lipids, though they are technically called as lipids. The right hand side you can see here, these are the fluorescent images of a planktonic diatom, and the yellow fluorescence that you see here are the TAGs. TAGs are nothing but neutral lipids. So it secretes enormous amount of neutral lipids to make it afloat all through this water currents. Whenever it is alive, it will be afloat. Once it dies or its lifetime is over, it sinks to the bottom, and it becomes the part of the food web, and also it becomes the organic matter and dead organic matter. And it is also known to be having this, because these oils are in humongous amounts. These are known to form the basic or the precursors of present days petroleum as well. So this is one particular evolutionary adaptation that diatoms have, especially these planktonic diatoms have acquired over time. And moving on to benthic diatoms, as I was telling, there are two different major categories. One is the planktonic diatoms. We have seen what a planktonic diatom is, how it is drifting through the water column, and what is the evolutionary adaptation it has. Now, moving on to benthic diatoms, these also have a different evolutionary adaptation. So benthic diatoms, when I tell this, so benthic diatoms does not do not float. So it tends to go down and then form colonies on any of the substrate that it comes across. So here you can see Synandra species that is forming a colony. So the colony can be patterned like this in Fragillaria species. You can see here, this is a joint side by side. It is a linearly arranged fashion. It looks very eye appealing, right? Sometimes it is very zigzag also. So you can see here, the Synandra species is clumped in a microalgae, green microalgae. This is the green filamentous microalgae on which it is colonizing. Okay, whatever substrata it comes across, it just holds on to it, and then it forms a colony. And the colony can be linearly fashioned, or this can be star shaped also. So different fashions it forms. So this is an Asteronella species. So it forms a star shaped structure, so while forming colonies. And this is another typical diatom, just Tabularia species. It forms it joins only towards the nodes, and then it forms a zigzag pattern. So for seeing also, it looks very eye appealing. So this is about benthic diatoms and how it forms, and then how it forms colonies. And another peculiar aspect of this benthic diatom is the EPS secretion. What is EPS? It is the extracellular polymeric substances. It is a carbohydrate polysaccharide that is secreted by these diatoms to form the colonies and be intact. So any substrata in the river, you say in an estuary or an open ocean, in any substrata that it comes across, it just starts forming colonies. So when rugged or a very forceful water current comes, it should not get washed away, right? So in temple ponds also, you would have seen a mat of green or yellow brown pigments that are there. So these are all this diatoms only, diatoms and the mixture of other periphyton mats, which consists of other microalgae as well. So you would have seen the floor is very slippery when these diatoms are there. This is because of this transparent structures that it secretes. So they are nothing but the EPS, so extracellular polymeric substances. So it also has a fashion or a pattern to it. So this is a Symbala structure. It forms stacks. as if you can see it is in the tree stalk, so or a branch kind. So it is very orderly fashion like this, or it is very random. So it forms like tube like colonies and then it attached to it. So you can see here in the third image, that is like a bunch of a flower. So these are like petals, and then in the center you can find this transparent structures. These are all this EPS. And the next thing is that it can form stacks or pad-like structures like this, or it can form a tube-like EPS, wherein inside it goes inside the EPS and then start surviving. So it will not come out. So this is again a mechanism that is that it has evolutionarily adapted to not being washed by this turbulent waters when they are forming establishing colonies in any of the substrata. Okay, so this is a peculiar feature of the benthic diatom. And coming on to this next interesting aspect of evolutionary, how this diatoms came into existence. So we all know the process of the aspect of symbiosis. So there is this symbiotic relationship, the well-known symbiotic relationship. If I'm to quote an example, this is between the plants, that leguminous plants, and the nitrogen fixing bacteria. We call it as in the root nodules there are rhizobium bacteria. This have a mutualistic understanding, and then it will not harm each other. So symbiosis means it will not harm each other. Instead, it will have a mutualistic beneficial attributes to it. So that is what is called as symbiosis. So when you talk about diatoms, this diatoms, many scientists hypothesize that this diatoms are the product of secondary endosymbiosis. So meaning that it has acquired, they cannot like circumnavigate or what categorize this diatoms into one particular species. It possesses the characteristics of many different species. So they are hypothesizing or postulating many theories. So one such theory is that these are the product of secondary endosymbiosis. So what it means is that, so heterotrophic host, when a heterotrophic host engulfed a green algae, which is again a product of primary endosymbiosis. So it is formed the primary endosymbiosis, it is formed because of the heterotrophic host cell engulfing a cyanobacterium. And then in the process, as and when the generations pass on, first it will be a symbiotic relationship between them, and later on the gene will get imbibed through the to the host chromosome itself. So this is EGT, that external gene transfer. Gene loss also happens. It is not that only all the genes get transferred. Some beneficial aspects of this the organism that entered the host, that is taken forward in the subsequent generations. That becomes a brand new species itself. So that is why you can see here the red algae is a product of primary endosymbiosis of a heterotrophic host as well as a cyanobacteria. So that is how this red algae is formed, is what the scientists are postulating. But the diatoms are a product of secondary endosymbiosis. That said, the heterotrophic host cell is engulfing a green algae. You can see it in here. So this picture is very self-explanatory. So it is engulfing the heterotrophic host cell is engulfing a green algae. In turn, this it is also getting the red algae is also getting engulfed. That is why you can see a like very complex and evolutionarily like higher organism like this diatoms. It has formed. You cannot put it into any particular category. That is why a brand new category is given. Family is it is put under a different family in itself. So that is how the diatoms have possessed the ability to both take up the nutrients and grow. When there is an unfavorable condition, there is no proper solar radiation is there, it switches to the heterotrophic mode, and then it consumes food as a bacteria does. So bacteria cannot synthesize its own food. Instead, it depends on the other organisms or the dead cells for their food. So that it switches that mode, and then when the favorable conditions turns up, it becomes a photosynthesizing organism again. So this is a peculiar feature of diatoms that you cannot see in any other organism. So, and also I was telling that how diatoms came into existence. So it came into existence a long back. So there are some hypotheses that tells that when the earth was not conducive for other organisms to live. So you know the theory of how earth is formed, how earth was formed. So when the earth was not livable for this other organisms at that time, because of the excess amount of CO2 was there. So this CO2 made the earth becomes a speck of dust, and then it was not conducive for the other organism that requires oxygen to live. That is why that is when this diatoms arose to prominence. So the ecological prominence of diatoms came into existence only during this time, and then it rapidly it was hypothesized that it rapidly proliferated, and because of this proliferation in the oceans, and the ocean surface, you know, it is 79% of the earth is oceans, right? So you can imagine how much of diatom proliferation has happened. Because of that, the CO2 content or the CO2 level of this earth drastically reduced, and in the process, so because the CO2 is getting decreased, so based on the decreasing levels of CO2 in those era, it was told that the diversification of these diatoms from radial centrics to rapid penates, these are all the different morphological forms of diatoms. These also came into existence because of the decreasing levels of CO2 in the atmosphere. So that being said, after this CO2 got reduced because of this rapid proliferation of these diatoms in the world oceans, so rapid cooling, global cooling happened. So because of that, the life forms in the oceans came into existence, is what the theory says. So even now some scientists believe that these diatoms are the only progenitor elements which was vital in making the earth livable. So this is one particular important aspect of diatoms that you should be knowing about diatoms. So that ends my introduction part of what diatoms are, how morphologically it is varied, and then what are the specific and peculiar aspects of diatom. Moving on, I will take you through this sampling, how if diatom, if you have a water body nearby and if you are willing to take up projects on this, how you can go about sampling. So as I was telling, it exists in both planktonic and benthic forms. In planktonic forms, you can see here that a reddish brown or orangish golden brown pigments. So these are the key that diatoms are present in there. So if it is completely green matte, you can like expect a less of diatom population and more of a green algal population. So you should search for peculiar golden brown color on the or a periphyton matte on the substrata. So as you see, once you see that, so you have to go about collecting it, right? So you need a very basic instrument for that. You need a spatula or a brush to scrape this diatom off. And then if it is a planktonic form, you need a plankton net like this. A quantum of water sample has to be filtered through this, say to 50 to 100 liters has to be filtered to get a decent amount of diatoms that you can like enumerate and then identify as well. So once that is and then it has to be collected in a any container, a non-reactive container, and you have to take it to the laboratory. And you can see here, if you directly observe under microscope, these are the variants of diatoms that you can see in 1 microliter of the sample. So it has huge amount of diatoms in 1 microliter itself. So there is a saying that if you take 1 spoonful of ocean water, and you can see millions of these organisms, but the only thing is that it is not visible to the naked eye because it is microscopic. And also, coming on to the processing, as I was mentioning before, when I was talking about the morphology and the intricate cell structures, I said striae and the puncta. These are the very vital factors that decides the which diatom, which genus or the species it belongs to. So, but what happens is that in the live diatoms, that organic compounds or the carotenoid pigments and the other pigments that are present in the chloroplast and other pigments that are present inside the diatom cells hinders the visibility. That is why you have to go for acid digestion. And also this is not possible in any other organisms except for diatoms. Why? Because other organisms like green microalgae for that matter, you cannot subject it to acid digestion. Because why? Because their cell walls are rigid, but it is made up of peptidoglycan. Peptidoglycan is again a complex polysaccharide. So as you treat it with acids, they are going to dissolve, and anyway they are going to the cell lysis happens, and you cannot do anything about it. But one peculiar aspect is that silica is inert and non-destructible. So that is the one particular reason that you can go for acid digestion. Once you digest this, you get a white precipitate that you have to neutralize it, because you are subjecting it to acid. You have to neutralize it, and you have to observe under microscope for exact identification or taxonomical classification. If you want to do, you have to go for this acid digestion process. And then that is all about the diatom sampling. I'm giving an overall view of, because in the interest of time, I'm giving an overall view of how is it possible, and what are all the aspects as well. I'm covering just the aspects. If you want in detail, you can go back and refer to any of the diatom books as well. There are many keys also on how you have to go about the manuals, also how you have to go about identification of these diatoms if you are interested in future. And coming about this diatom blooms. So nowadays it is very prevalent that our coastal regions, even in the Arabian Sea, we are facing this algal blooms. So, but diatom blooms is not predominant in this in our region, tropical region, but it is predominant in like temperate regions like Europe and North America. So every year, during the spring season or the winter season, as and when the onset of winter season, they tend to have this big diatom blooms. This is causing a menace for them. Diatom blooms are menace everywhere. So here also in Arabian Sea, we are facing the dinoflagellate bloom. As and when in Carver, you would have seen the green tide, and in Japan the red tide. All these are the results of the anthropogenic pollution, excess amount of anthropogenic pollution that we are subjecting these coastal regions to. So that is why this diatom blooms happen. And as and when the diatoms bloom happen, this create obnoxious odor, and also it is known to have detrimental effects on the coastal ecosystem, because the balance in the coastal ecosystem is completely hampered because of this. So fish death and other unwanted effects also happens. So this you can see here, it is a diatom bloom that has happened in Europe in 2015. So this is a news article that came during 2015, that record-breaking toxic algal bloom of the west coast in the Pacific Ocean. So you can see here, this is an Alaska coast in the North America. In 2015, it has got this Pseudoneschia species, that diatom bloom. This Pseudoneschia species is known to produce a toxic neurotoxic substance. So that is called as domatic acid. So this domatic acid, what it does, it will stun the brain effects for a brief period of time. It makes the animals which is consuming this organism, and also it even affects the human health as well. So if the fish which has consumed this, if a human is consuming that fish, you also will have that major neurological effects in the long run if you keep consuming it, because biomagnification is at a higher level when it reaches the humans. So this is one particular havoc that the diatom bloom is creating across the world oceans. So as I was telling, this is the Pseudoneschia species, which can be categorized by it is like a normal Neschia species, but at the ends it forms colonies by attaching its end to the other one. So it forms a long needle-like structure. So this is a peculiar characteristic of Pseudoneschia species, and it also affects the clams. If clams are known as filter feeders, so if this Pseudoneschia bloom happens, and if the clams happen to ingest that, it stays there for a longer period of of time, and then if the livestock comes into contact and it drinks water in the regions also, it tends to get sick because of this diatom bloom. So we have to be careful, and this shows the diatoms blooms are nothing but an alarming signal that we have to reduce the coastal pollution, and then be wise in reducing the pollution, and then taking decisions about what has to be let into the ocean, and how we are where are we in terms of protecting the environment. And moving forward, this is a glimpse of how diatoms are becoming, or it has been from 19th century. It has been used as a major tool for as a bioindicator. So it is not only the species. There are other algae also. Even in Asulaba's webinar talk, you would have seen algal species also acts as a bioindicators. So there are keystone species in every ecological landscape. So those becomes a bioindicators. If you want to know about the serenity or the balance in the ecosystem, you have to understand this species. If a species is not present or the diversity is hampered, you can know that there is some kind of pollution that is happening. So say for example, in terms of diatoms, if I talk about, so Cyclotella species, this is known to proliferate in sewage maturation ponds. So in sewage maturation ponds, if there is a water body nearby and suddenly there is something happening, so some obnoxious smell starts coming, you want to understand the quality of that water body nearby, you have to just take the water sample and analyze for this diatom's presence or absence. So if Cyclotella species present, that means that the water body is like polluted by this sewage. So some domestic sewage is entering the water, that you can make out. So these are called as bioindicators or keystone species that for the identification of the pollution that we are causing. So say for example, amphora species. Amphora species thrives where there are solid waste, a lot of solid waste which has organic content to it. If an organic content is higher in any of the water bodies, if there is a hotel nearby and it is dumping all the organic waste into the water body nearby, so then amphora species proliferates. And also the same case with the Nischia species as well. And also in navicular species, these are known to be like having a higher tolerance to metal pollution. So if there is an industry nearby which is causing which is letting the like metal contaminants into the water body, so no other species thrives there except for this navicular species. This is known to have higher metal tolerance, so only this species thrives. So if these species are only present, then you can say that the source of pollution is from an industry which is having a high metal concentration. So like this, you can go about understanding the bioindicators and the level of pollution that is being caused because of this anthropogenic activities. So moving on, as I was telling, this is again the brief of what I was discussing earlier. So Cyclotella species, the species towards the left are the hardy species or the opportunistic species that can thrive in higher levels of pollution, wherein the species in the right hand side like Gomphonema, Alocosira, and Rhamphonus and Spinularia species, these species are called as sensitive species. So they cannot thrive in higher levels of pollution. So as and when there is a pollution, this species either goes off, dies, or it goes into dormancy. Till the favorable condition comes, it will not reproduce, or you will not be able to see this species in that particular water body. So that is how this bioindicator, diatoms as bioindicator works. So moving on, the bioindicator thing is for the present level of pollution in the lake. So if you want to, if you want to reiterate or reconstruct the climatological condition which was there some 1,000, 100 or 10,000 years back. Say this is a study that is carried out in Quebec Lake in Canada. So because I keep on telling that diatoms have a inert silica cell wall that is not destructible. I mean, when I say not destructible, it is for the ages. It cannot be destroyed. So it lies intact in the sediments. So what you have to do is that this climatologists, paleoclimatologists, what they do, they dig the core and then take out the sediments at various strata in the water body, and they try to analyze what was the species diversity that prevailed before, like a long time ago, say some 11,000 years ago. This graph is a typical example of that. Say from time zero, this is a time zero at the present condition. What is the diversity of the diatoms? These are benthic species and planktonic species. And what is the species composition as and when the ages dates back? So this is some 11,000 years ago. So they do the dating studies wherein they understand, they try to understand what was the level of nutrients at that time, and what was that kind of species that was present during 11th before 11,000 years ago. How was the lake before 11,000 years ago? For that, paleoclimatological studies also, diatoms are like useful. So this is a statistical analysis that shows how the physicochemical parameters of that lake through the ages has helped in the diatom species formation of the community. So this graph shows that. So this is a glimpse of diatoms along the east and west coast of India. So this is a secondary data collated by me. So this shows what are the different kinds of benthic organisms, benthic diatoms that are predominantly present along the coast. So coast, when I mean coast, it includes estuarine areas also, estuarine and the coastal river mouth as well as the coast. So these are the some of the species that are present in along the east and west coast of India. So that ends my topic on diatom as from an ecological perspective. So moving on, I will talk about the beneficial aspects from an industrial relevance or the economic perspective. So the first and the foremost in that economic perspective is that, which is known from centuries old. This like um, like this aspect of diatoms being used as a live feed is a century old. So from the 18th century onwards, people started using this as an aquaculture feed. So first and foremost reason why diatoms are preferred sources of feeds. When I say live feeds, you know aquaculture practices at a large scale. So when they are growing fishes, so they cannot feed them with the juvenile fishes, they cannot feed them with any other organisms but for this diatoms, because they are very small. Their mouth size is very small. They have to have this planktons as in the natural ecosystem. You have to replicate the same condition in the like lab as well. So when the aquaculture practice is going on, you have to feed them with the very favored source of food for this for the juvenile fishes. That is why that is how this came into existence. So diatoms are being used as a live feeds. So in India, CMFRI is like leading producers of this planktonic species as well as the benthic species for the as a live feed for juvenile fishes. So not only the juvenile fishes, even the big fishes also are fed sometimes because of the presence of omega-3 fatty acids. So diatoms are known to be rich in omega-3 fatty acids. It's like EPA and DHA. So what are these essential fatty acids is that humans don't have the tendency to produce or synthesize this essential fatty acids, but in turn these are very much required for our a proper functioning of the brain. So like DHA is very much needed for any even the small children for their brain growth. So you would have seen Horlicks advertising for this DHA and EPA. So these are essential fatty acids which are high value molecules. So these comes in a like very small quantities in this in diatoms. So diatoms are known to be rich source of omega-3 fatty acids as well as this DHA. We consume fish, a lot of fish, considering that it is a source of omega-3 fatty acid. But the actual fact is that because fishes consume a lot of these diatoms are precursors or the basic source of EPAs or the DHAs. So because it consumes a lot of it, so when it is a bioaccumulation that is happening, that is how the fishes are getting the DHA as a source of it, so that we are consuming it in turn. But the basic source of these omega-3 fatty acids are these diatoms. So having said that you can understand the importance of these diatoms as a live feed. Moving on, The next aspect of diatom is that I had mentioned it earlier also, that the present day's petroleum is because of the humongous amounts of diatoms that has been formed before ages, and then it sank into the ocean floor, and it forms because it had lipids intact. So those are the ones in the ocean floor we dig for petroleum sources, right? So the finest quality of petrol, when they did the tracing studies or the biomarker studies, they found that the stearins that are present in the diatom lipids that are very much matching with the present day's petroleum. So you can understand that how diatoms form the basic resource that we are exploiting at a larger scale, how it has come from. So these are very minute and microscopic organisms when we see about, but the quantum of the diatoms that has gone into the ocean, and then we are getting it in terms of petroleum, and we are running it like from us, and when the industrialization started, we are consuming it at a larger scale, this petroleum products as a larger scale, and we say that it has stocks as well for the future as well. So ocean floors are the major sources of this petroleum stocks, and you can see that diatoms are these microscopic creatures are the like costs or the stocks for it. So you can see the importance of this diatoms in our present times as well. It has higher relevance. Diatoms have higher relevance in our for our present generations. Maybe the consumeristic attitude as well as the industrial exploitation that we are moving forward. Also, it has higher relevance. And the other aspect of diatoms is that it is acting as a third generation biofuel. So now recently in the after 1900s they started seeing diatoms as a precursor or the major sources of feedstock for biofuel production. So as I was telling, that petroleum feedstocks itself has come from this diatoms. Then why not take this live diatoms and extract the lipids, and then which are having a higher biodiesel quality. So the lipids are of higher quality that can be used as a biofuel itself. So that is when with that context only it started in the 1900s. So even now people are looking into the aspects of it, but the only thing is that how to make it economically feasible is what the scientists are looking into. So the left side, the flow chart talks about how it can be how the biodiesel can be extracted. It starts from the strain selection, or they have to culture the diatoms, and you have to, once the diatoms reaches the stationary phase, you have to lyse the cell and extract the lipid, and then lipids can be converted into biodiesel through the process of transesterification. So in the right side column, this is the process that is involved in converting a diatom, live diatom, into biodiesel. So the lipids are to be extracted, and because it is not being economically viable, now scientists are looking into how the other left out products also, like carbohydrates. It has a major portion of carbohydrates as well as proteins, how it can also be exploited for other value added products other than this biodiesel is what now people are looking into. Even my PhD work is on extracting lipids from diatoms and in turn converting that into biodiesel. So this ASP and diatoms, I want to tell how important the diatoms are in terms of third generation feedstock. This ASP is a program that is conducted by the Department of Energy by the United States. So it started in 1985 and it ended in 1998. So they did a rigorous screening of all this microalgae, and then they saw which all the species which has a potential as a third generation biodiesel feedstock. So the results states that they, out of 3,000 species, they narrowed down only to 50 strains. So those 50 strains also, 60% of the 50 strains that they have chosen as a potential feedstock are diatoms only. This is peculiarly because of their higher growth rates and the high quality lipid that they secrete. When you consider the cell volume to the amount of lipid that it secretes, the diatoms are the ones that outcompete other forms of microalgae. So when you consider green microalgae and cyanobacteria, they have a lesser volume of lipid content when compared to these diatoms. That is why diatoms are well known or getting popular as the third generation biodiesel feedstock. So this is a completely different perspective of diatom. So diatoms are useful in forensic sciences as well. So this is not an industrial exploitation. It doesn't have an economic value to it, but it is very useful for the experts to understand whether if any crime scene happens or any death happens. So if it is a suicide or an attempted murder, so if they want to understand that, for that also diatoms are useful. See the pace of diatoms and the usefulness of diatoms and the diverse arena. So how it works is that, as I was telling, silica is non-destructible and it is inert. So when a person accidentally fall into the water body, what happens if he is accidentally falling, or it is he is attempting a suicide? So he would obviously engulf some of the water, so that reaches the lungs. As and when the lungs are getting filled with water, and it slowly the water gets pumped, because the heart also keeps functioning, right, before he dies. So the heart pumps all this water into the other tissues as well, so the bones also. So the diatom reaches till the bones if a person is has accidentally fallen into the water, or he has attempted suicide. So intentionally if he has fallen, this is bound to happen. So it reaches the tissues. So what they do, if a person is drowned in the water, they check for this diatom's availability or the presence in their tissues. So this portion you can see here, this is the region that it is called sternum. Sternum is a region that connects the upper thorax to the lungs. So it first, the diatoms enter into the water has diatoms. So when the person is engulfing the water, it goes into this sternum, so and it reaches this lungs. So any part of the lungs tissues from the lungs or the sternum regions are taken for during autopsy. They cut this body parts of the deceased person, and then they go for tissue digestion, and then tissues get dissolved, but not the silica frustules. So silica frustules will be intact. They go for a microscopic examination of these diatoms, how diverse, what is the diversity that is present there, and then they go back to the water body and take water sample and also analyze if the diatom diversity in the human body as well as the water matches. Then that becomes a accidental fall or a suicide. In turn, if a person is murdered and then put into the water body, the water will enter, but not to the extent of till the bones or the tissues. So if the person is already dead, the dead person will not breathe, try to breathe or engulf the water to that extent. So that is then if they analyze the tissue, there will not be any diatoms, so they can understand that this is a murder. So this is one particular peculiar aspect of diatoms in the forensic sciences, you can say. And moving on, now this is a very nascent field wherein diatoms are being targeted as a drug delivery system also. So the diatoms, as I was telling, it has a very amazing nanostructures. You can see here, it has very intricate patterns of pores that are present. So it can be used as a biocompatible as well as a cheap source of nanomaterials. Otherwise, the synthetic nanomaterials are very costly to manufacture or synthesize itself. So why not use these diatoms? These diatoms have got very patterned structures into it. So based on the diversity, based on the species that you are opting, so it has a very diverse and intricate structures of this striae or punctas. So the punctas are pores, right? So what they do, they take certain concentrations of this medicine, and then they impregnate that with to this pores. So that becomes a targeted drug delivery system, that if you can take it as an oral consumption, as well as you can go for targeted delivery, drug delivery also. In some of the studies in mice, they have done that anti-cancerous property. So anti-cancerous drug, what they do, it is very specific. Cancer is very specific, right? You have throat cancer, you have skin cancer. So if a person is suffering from skin cancer, what they do, instead of taking orally, they inject that drug which is impregnated into this diatom cells, into the targeted body parts. So if it is a skin or if it is a throat, you have to inject that into that particular body part, wherein it has a sustained release of this drugs for a prolonged period of time. In mice, the study was carried out in which this anti-cancerous drug, which is impregnated into this cells, were given to its skin, and it was found that it had a sustained release for a nearly 30 days of time. So that being said, it is very highly sought after. It is becoming a sought after component of the drug delivery, recent drug delivery systems also. So high end sophisticated drug delivery systems also. So this is another aspect of diatom. And moving on, this is the bioactive compounds. I have talked enough about it. So it has many biochemical components. So one such component is carotenoids, as I have already spoken about it. Why it is important? It is useful in the cosmetic industry. And otherwise, you have sugars and you have lipids and you have pigments, other pigments other than this carotenoids as well. So the major use of any pigments is that it acts as an antioxidant. Antioxidant is a property which reduces the risk of cancer. So when reactive oxygen species forms in our body, that is called as tumor cells that aggressively proliferate, and it cannot be stopped. That is why doctors suggest us to take green tea. Green tea is full of antioxidants. So antioxidants will reduce the risk of cancer in any patients or any human beings as such. So that is why this pigments are becoming most important, and it is playing a vital role in industrial sector nowadays. So that is where the large scale cultivation of one particular diatoms or microalgae just for the extraction of these pigments are happening. So once the pigments are extracted, it is made into powders, and then you can take it as a tablet form also. Otherwise, they can put it in creams also. And then, as I was telling, that antioxidants can be used as in powders, directly as powders, or as medicine, or as in UV, anti-UV or UV protective layers also, and creams. So this is one particular commercial product which is sold as a anti-pimple powder. So this is a very less important when compared to other pharmaceutical application, but still it has its own application at various levels. So this is another set of products that are purely made of microalgae, especially diatoms. So you can see it has industrial aspects as well as higher potential towards the high value molecules also, rather than it is being used as a feedstock for biofuel production. From the energy aspect as well as the high value molecules, these diatoms are most sought after. So the other aspect of diatom is the solar panels, diatom solar panels. So the very pioneering idea of this diatoms being used as a solar panel came from my mentor, our research group, in the form of paper. That is most read article, milking diatoms. So the idea of this is that, so why we do not cut the cows for milking it. So we just milk the cows, right? So the same way, the diatoms are also having oil. Why not we milk or milk the oil into it, from it, by biochemical engineering the diatoms. When you grow the diatoms for a particular period of time, after it should ooze out the oil in itself. So why not you go for a biochemical engineering aspect of it, so that you form a diatom solar panel. It starts to proliferate. Once it reaches a certain stage, it has to ooze out the oil. The oil can be extracted and it can be used for biofuel production also. That idea came from Dr. TVR. So in his paper, he has quoted this, and based on that, many recent works have been done on these aspects. So diatom solar panels are getting into the research, if not in the market as such, commercial market as such, it is being researched widely. How this silica content. So the solar panels are made of silica. So when silica is doped with any of the d block elements like arsenic or germanium, it becomes a semiconductor. That semiconductor can be used as a for converting the solar energy directly into electricity. That is what the panels does. PV panels do. So why don't we, instead of going for synthesizing silica, which is again a costly affair, you go for a biocompatible silica. You grow diatoms, which is very cheap, and then it has nutrients in it. We have abundant amount of wastewater, so a wastewater can be instead be used as a nutrient, and then grow these diatoms. And then once the diatom cells grow, you just take the silica and use that as a parent element or the progenitor element for this semiconductors, for the production of semiconductors. Why don't we do that? So what they do, they impregnate, or they forcefully they metabolize this diatom cells to take out this TiO2. TiO2 is a titanium dioxide. This is a molecule, it is a metal that when it imbibes that, when diatoms grow, it tends to accumulate all the metals that comes into its contact. So it absorbs this molecule, and then it acts as a semiconductor. What material which was inert before, it has become semiconductor just because of the ingestion of this TiO2. Why can't we use that? That kind of aspects also are being looked after for this diatoms. And also, as I was telling, the another aspect is that without lysing the cell, so how can you extract oil out of it? So this is one typical study wherein they are trying to see how a mechanical pressure or an osmotic stress or an electric shock wave for a brief period of time can be given, so that the diatom cells cleave, and then it oozes out the oil. You can see here in this photograph, the lipid droplets is oozing out of the cell. So why don't you cleave that and then make it happen, and then it regains its forms, cell form, because it is not lysed completely. The other components, chloroplast components, are intact. Well, it will regain its viability in a brief period of time. Why don't we look into that? That kind of aspects also are being looked into it. Without lysing the cells, how can we reuse it and then make it more effective than making the batch cultures every time and lysing the cells and then getting the lipids out of it? That kinds of study also is widely looked into in recent times. So the last aspect I would want to talk for today is that diatomaceous earth. So the previous topics and all are how the live diatoms, and what are the importance of live diatoms, and what are of its cell components. So when the diatoms die, it goes to the ocean floor, as I was telling. So it came into ecological prominence a long back, and then the ocean floors are full of this diatomaceous earth. So reports say that up to 300 feet down the ocean floor, you get this diatomaceous earth. So you can see that the amount of diatomaceous earth that we are left with. So why don't we use that as for industrial exploitation also? People have looked into it, and because of this nanostructured silicate ornate cell arrangements, it has very intricate structures, wherein you can see here there is a big pore, and inside that there are many minute pores. These are the defensive mechanisms that these diatoms have formed, such that they are not affected by this viruses or bacteria of very smaller shapes will not enter into this cell membrane. But when the cells die, this can be used as for the same purpose also. So when water has to be purified, it is mainly used in water purification systems. If the diatomaceous earth is packed in the water purifier, and water is let go of into this systems, so what it does, it filters out all the bigger particles like dust, as well as the bacteria. If there are any bacteria that are viable, that also is get filtered out, and then you get pure water. So for that aspect, this diatomaceous earth is being used. This is an industrial setup wherein the diatomaceous earth are being used as a water purification system. And apart from that, you have other aspects of diatomaceous earth as well. These are used as fillers in the production of talcum powders, and also there are food grade diatomaceous earth that are available. So when you eat that, they say that it acts as a cholesterol regulator, and then for if obese people are there, they can intake that, and so that the diatomaceous earth acts as a cholesterol regulator and reduces the amount of cholesterol that is present in the body, and also it is used for preventing constipation also. And then it has aspects of other dimensions of giving hair and bone health. If you want to maintain that, there are you can use this as a like you would have seen pumice stones and such kinds. So these are called as abrasives. So the abrasives. So that has a property to remove the dead cells from our body, even the bones and the hair which is unwanted hair. If you want to remove, you can use that as an abrasive. And also the diatomaceous earth are known to provide ovothiol. Ovothiol is again an antioxidant which has anti-cancerous property that is generated from this diatomaceous earth also. So that is another aspect of this diatomaceous earth. So some miscellaneous aspects of diatomaceous earth is that this is my last slide. I'm going to end my talk with this. So food grade diatomate that I have already covered. Apart from that, it has insecticidal properties also. If you want a non-toxic method of getting rid of your garden pest, you can use this. How it works is that when diatomaceous earth are put into your over the garden pest, it desiccate the pest, meaning it will absorb all the moisture content and including the white blood, because insects, they have white blood, right? It doesn't have a blood as such. So all that moisture content it will remove, and it slowly lyse the cells. So that is how it can be used as a organic way of insecticide also. And the last property that I would like to discuss is that dynamite. Dynamite, it is an explosive. It was invented by Alfred Nobel. So what happened was Alfred Nobel was trying to make an explosive that is stable at room temperature. So this this trinitrogen, that is that is an explosive which is not stable at room temperature. So when it is mixed with this diatomaceous earth, it became a stable compound. That is how an explosive was invented by Alfred Nobel. So that is the last, but no, it is very trivial use, but in military applications it has its own use as well. So that ends my talk on the diverse aspects of diatom. But I'm saying that it is not an exhaustive list of all the aspects of diatom. There are n number of other aspects as well which I have not covered. But nevertheless, these are you can see that the dimension in which the diatoms can be useful is enormous. There is an enormous perspective of this particular species. So indeed, I hope that I have justified my initial slide that I was telling, if there is wonder in this world that is contained in water. So that indeed is very apt statement that Lauren LC has made. So with this, I would like to conclude this talk, and thank you everyone for your patient listening. And I end my talk. If there are any questions, I'd be more than happy to answer. So with this, I hand over the session to Deepthi. Thank you. Deepthi, your voice is not audible. Yes.
Am I audible?
Yes.
Yeah. Thank you, Sharanya, for the wonderful talk. So there are a few questions. I'll just read out them to you. The first
Yes.
question is by Shukla. He is asking, is there any quantification data that how much diatoms consume dissolved oxygen with respect to their living pattern in different forms?
Okay. Consume dissolved oxygen, is it? I don't think.
No, it is based on the carbon sequestered. This is the quantum of carbon stored in the atom gives the indication of the oxygen.
Okay, but it doesn't consume any dissolved oxygen as such, but it liberates oxygen. It is vital for the presence of oxygen in any water body. It depends on the rate of carbon sequestration that diatoms undergo or diatoms are able to do. How much of carbon it can fix, that is equivalent to the amount of dissolved oxygen present in the water body. It is a proxy that we can use. But as such, diatoms are not involved in the consumption of dissolved oxygen.
So the next question is by So the next question is by Riju. He is asking, since diatoms have hairy structure and form colony as a defense mechanism, are there any organism that has adapted itself to feed on diatoms?
Yes, in spite of all these adaptations, there are organisms, even the Chitoceras species which I was talking about, they have the flagella, pseudoflagella, as I was telling, right? But instead, these Chitoceras species are the most fond or the sought species of this bigger juveniles. Even in CMFRI, Chitoceras is largely cultivated just for the as a food for this sea bass. Sea bass is a cultured species, fish species in CMFRI that consumes this Chitoceras species. That is the most preferred food for this sea bass and other, not only sea bass, other big fishes also. It is a very preferred source of like food. Yes.
So the next question is by Shukla again. He asks, can we do most probable number, that is MPN test, using diatom as bioindicator, just like E. coli test? Is there any number that indicates?
Yes. Number is actually there, but it is not the most probable numbers. That is, bacteria are even the lesser strata of species. We go for most probable number, but these are higher organisms which are of the size 20 to 200 micron sizes. So the most probable number is only for the bacterial species. And but if it comes to diatoms, there are indices. There are diatoms indices available based on what is the number of species. Based on the species they are categorized. So if species diatom indices is so much, there is a numerical value to it. So if it is from 1 to 10, it is not an actual indices, but for as such, but for an example I'm telling you, if it is 1 to 10, that says that what is the extent of pollution that is happening in this water body. If it is an oligotrophic lake, the diatom indices should be at a higher level, because the diversity is more. If it is an indices is reducing to from 1 to 10, if it is very less, that means that the water body is highly polluted. So that kind of indices are there in terms of numbers, but it is not in terms of most probable number.
So the next question is by Rashmi. She's asking, this is regarding the diatom that you mentioned from Alaska coast. How can people avoid the growth of such toxic diatoms?
It's as simple as that. You control the amount of pollution that you are creating to the environment. If directly the industrial effluents, it is a major cause of the anthropogenic disturbance is a major cause of any diatom blooms. If you are controlling that, there are some aspects which are climatological conditions also. When upwelling happens, sediment upwelling happens, all that thermal stratification happens in the colder regions, then diatom blooms happen. So then also you have to understand and then take proper remedial measures. What is the source of pollution, and then you curtail the source of pollution so that the diatom blooms is avoided.
So the next question is by Manoj. He's asking, are we doing any Bayesian analysis for Bangalore lakes with regard to benthic diatoms diversity?
I haven't worked on freshwater systems.
It is done by our group.
Okay, but yes, Sulabha and Cincy have worked on this aspect. But what is that Bayesian thing? Is it? Can you repeat that question?
Bayesian analysis.
Bayesian analysis, is it?
Yes.
Okay, species diversity analysis and indices and all, it's been calculated, but Bayesian analysis, I'm not sure. Asulabha and Cincy are the right person to ask this question, so they would be able to answer this.
Those are the questions that we have received.
Okay.
Are there any more questions? Kindly post in the chat box so that we can address them. Since there is a delay.
What was the question? Bayesian?
Bayesian analysis, it seems.
That is just a fuzzy result.
Okay.
It's a soft result. I don't know why it's a Bayesian analysis. Any analysis.
Okay.
When you calculate using the data, it becomes a hard classifier. When you go on with this is a soft classifier.
Okay. Okay.
So I think there are no more questions, Sharanya. I think we can conclude.
Wind up the session. Okay.
So we are winding up the webinar for today. So I would like to thank all the participants who have actively took part in this webinar and have asked really good questions. And then also I would like to thank Sharanya for taking us through the diatoms of the ocean and making us introduce to the different aspects of diatoms, from the commercial prospects to the pharmaceutical, cosmetics, all the different aspects. And then she even told about its usage in forensic science and drug delivery system. So this was a very interesting topic for today. And thank you so much, Shania, for taking part
Thanks,
in this.
Deepti. Thank you, Deepti. Thank you all again. It has been my pleasure to share the knowledge what I have acquired all these days on diatoms. So I thank Dr. T.V. Ramchandra for giving me this opportunity to share my knowledge with you all people. So thanks once again. Thank you so much. It has been pleasure delivering a talk to you. Thank you.